Today we have the pleasure of hosting Dr. Anne-Marie Schmidt for a talk on obesity and diabetes. Dr. Schmidt is a professor of medicine, pharmacology, and pathology at NYU. She received her medical degree with honors from NYU, where she later completed her medical residency, chief residency, and fellowship in hematology and oncology. She then moved to Columbia University, where she joined the Department of Physiology and Cellular Biophysics as a postdoctoral fellow of the Juvenile Diabetes Research Foundation. From 2003 to 2010, she served as the chief of the Division of Surgical Science and the Gerald and Janet Harris Professor of Surgical Science. In 2010, Dr. Schmidt returned to NYU as the director of the Diabetes Research Program. Dr. Schmidt's laboratory discovered RAGE, the receptor for advanced glycation end products, a cell surface receptor that exacerbates inflammation and damage when activated. Her laboratory has recently discovered the intracellular interactions of the RAGE cytoplasmic domain with DIAPH1, a member of the Forman family. This discovery forms the basis for the identification of a new class of RAGE inhibitors for the treatment of diabetes and its complications. Please join me in welcoming Dr. Schmidt.
Thank you very much. Good morning, everyone. It's very nice to be back to Mount Sinai. I've come here a few times to give lectures, and it's always very, very nice to be here. So this morning, I wanted to tell you about some updates with the RAGE story, fill everyone in on some of the basic background, but also give you some of the sense of where we are moving and how we think this receptor is potentially targetable for therapeutics. So the receptor was discovered now in quite a number of years ago, actually in 1990. And the initial context in which we were working was to try to understand how the advanced glycation end products, which are the products of non-enzymatic glycation and oxidation of proteins and lipids, whether or not these ages, especially in diabetes and also in aging, whether they were merely biomarkers of the metabolic change state, or whether in fact they could be contributors to the pathogenic mechanisms. And so the strategy that we took is we knew that ages could bind to cultured endothelial cells in a dose-dependent, saturable manner. And eventually we used large batches of endothelial cells with a radioligand binding assay with I-125 labeled H protein in order to be able to try to fish out a binding molecule. And it was pretty exciting, you know, going down on these gels when first there were like too many bands to count, and then down, 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 until finally the one band went for sequencing and had an identity that had yet to ever be identified. And this turned out to be an immunoglobulin superfamily molecule, which already put this as something really super exciting. Because one of the possibilities, of course, is that this could have been a typical type of scavenger receptor molecule, but its presence in the IG superfamily really gave it the possibility to be very distinct in terms of the mechanisms by which it might operate in diabetes, and as we found over the years, in other states as well. So one of the first tasks then was to make an antibody, and we discovered right away that the receptor was expressed in many different types of cells. Generally in homeostasis and post-development, the expression of the receptor is quite low in human and rodent tissue, also in swine. And with diabetes and with immune inflammatory activation, the expression of the receptor increases in immune cells, vascular cells, and very interestingly also in cells such as the podocytes in the kidney and cardiomyocytes in neurons. And these are cells, of course, that are targeted for dysfunction and diabetic complications. What also was very interesting is that the highest expression of the receptor, even in homeostasis, is in the lung. And I'm very excited that over the years, so many people have asked me for the RAGE knockout mice who work in lung biology and pathobiology, and they have really made a beautiful story about how the RAGE molecule appears to be important in chronic lung disease. They can detect RAGE in this alveolar fluid that they can take from people as well as the animals, and it appears to be a biomarker. So I don't really talk about that work, to be honest. We haven't done that, but I'm just thrilled that others, you know, have taken that on and really looked at why the expression of the receptor may be high in the lung. And so the other component of IG superfamily molecules is that they have cytoplasmic domains They usually are very important for their signal transduction. It turned out, and we discovered years back, that the cytoplasmic domain of RAGE did not have endogenous tyrosine kinase activity. And so one of the challenges, which we believe we hopefully have solved, at least in part, is a cytoplasmic domain binding partner that we're really working very hard on trying to understand that relationship, whether or not it's mechanistically important, and of course, is it targetable. And over the years, the other very exciting thing about the RAGE pathway, so I told you that the original ligands were these advanced glycation end products, which are post-translational modifications of proteins. And it turned out that not unlike other Ig superfamily molecules, this receptor is a multi-ligand receptor, and other molecules such as the inflammatory HMGB1 and S100 calgranulins we identified as ligands for the receptor, and also oligomeric forms of A beta, which gives inference to the receptor in the pathogenesis of AD. And also others identified that phosphatidylserine is also a ligand for the receptor. So again, it's not too unusual in the Ig superfamily that this could be the case. And what we think that this relates to then is that there are multiple different situations that the ligands of the receptor accumulate in. And in many ways, it was always a challenge. Well, how could this be? How could all these different types of molecules bind to this receptor? And it turns out that others who solved the structure of the extracellular domain, because the ligands mostly bind at the V domain, understood that there were two major areas on the V domain where the ligands could bind. One is a so-called negatively charged area, and the other is a hydrophobic area. So in many ways, it does come together because of this versatility of the V domain. Might make it a little hard to target extracellularly if that's the case, and this is one of the reasons why our potential aims at the therapeutic approach are toward the tail. And so what are the disease settings and states in which these ligands increase in? I've already mentioned about natural aging and diabetes, Alzheimer's disease, of course, because of A beta, as well obesity, very recent work that we have done from our laboratory, and also obviously in diabetic complications such as atherosclerosis. So when the ligands bind to the receptor in the many different types of cells that I mentioned already, many labs now have shown, in addition to what we had originally described, is that RAGE appears to be a very potent mechanism for generation of ROS. It's very interesting, as you know, there's now this yin-yang story about NOX4 and whatnot, about good ROS and bad ROS, and this is something very important to think about, how nature puts things together. In general, what it appears to be, at least in terms of the RAGE pathway, is that the NADPH oxidases are a chief source of RAGE-driven ROS generation. We also are discovering through unbiased and also now very focused experiments roles for RAGE in lipid metabolism that we hadn't really expected. Also, RAGE is very important in generating cytokines and also in cellular migration. Finally, phagocytosis is also an important function of the receptor. And all of these things obviously are very intriguing because they have obviously normal homeostatic roles, but then obviously in disease settings, these can go awry and contribute to the pathogenesis of disease. So one of the challenges, of course, is to keep trying to figure out the natural function of the receptor and at the same time figure out how it can be targeted. So many years ago, after we identified the receptor, what our interest was in the macrovascular complications of diabetes. That's what stimulated the research. That's what triggered the search for potential receptors for these ages. And one of the very first things, as I mentioned, is we made that antibody, then shared that antibody with researchers who had access to the human samples. And essentially, in human atherosclerotic plaques, it was discovered quite a number of years ago that there was a greater expression of the RAGE molecule in the diabetic atherosclerotic patient versus the control patient. Interestingly, and not surprisingly, because of that gamut of ligands that I told you about, even in the non-diabetic state, RAGE is expressed in the atherosclerotic plaque, but certainly more so in the setting of diabetes. In experimental models, back when we first identified this, it was a bit challenging because of the lack of good mouse models of accelerated atherosclerosis. So just at the time that we became interested in this experimental question, Two independent papers were published by Nobuyo Maeda and Jan Breslow, where they described the ApoE null mouse, which was the first mouse to be identified that could develop spontaneous atherosclerosis even on a normal chow diet. Nobuyo Maeda shared those mice with us. We back-crossed them into black 6 and then rendered them diabetic with streptozotocin. We used streptozotocin because it was readily clear how to be able to induce hyperglycemia and still keep the animals alive. It was a balance of aiming for about 250 to 300 milligrams per deciliter. And we sustained the diabetes in the animals. And basically what we showed was that the acceleration of the hyperglycemia was in part also related to increases in the atherosclerotic lesion area in the mice. And so at the same time, we were generating our RAGE-deficient animals, and we bred the ApoE null mouse into the RAGE-deficient background. And basically what we found was that there was a very significant decrease in the atherosclerotic lesion area in the double knockout animals versus the single knockout. And importantly, the levels of glucose were the same in both groups of animals, so that the RAGE knockout for some reason was not immune to the effects of the streptozotocin. Back in those days, we used to use columns to actually measure mouse glycosylated hemoglobin, and they were absolutely identical. The lipid profile, the total cholesterol and triglycerides, was the same in both groups of mice. One of the things we question now is whether or not the nature of those lipids is in fact the same, and that's work that we're beginning now with collaborators to try to better understand. But at least in terms of total number, this was not changed. But what was changed was the content of the lesions. And in fact, what we found was that in general, when we deleted the receptor, there was less immune cell content, macrophages and T cells, but a greater content of collagen, which at least in mouse atherosclerosis is generally a marker of greater lesion stability. And so this work then has really challenged us over the years to try to figure out, what the critical cell types are that are mediating this. So as you know, in 2012, we had that terrible Sandy at NYU that just knocked out all of the brand new generated RAGE floxed mice. And so now these mice are back up and re-derived, and we're now doing the tissue-targeted work to try to understand what is the chief cell type in terms of the RAGE that's really mediating these very dramatic effects that appears in atherosclerosis and other diabetic complications. And so to make a long story short, over those very difficult 4 years, we used a lot of global RAGE knockout mice, we did a lot of bone marrow transplantation, and we isolated macrophages from the animals. And what we discovered initially through an unbiased array was a very exciting finding that in the RAGE-deficient macrophages, especially in the diabetic state, that there were higher levels of these cholesterol transporters, ABCA1 and ABCG1. We verified that then by real-time PCR, and essentially it was very important to note that just looking through the literature that it was already established in type 1 diabetic human and also in the mouse models that these transporters were negatively affected by the presence of diabetes. And in fact, these experiments suggested that perhaps the RAGE pathway might contribute to this. We also looked at the cholesterol efflux, and in parallel with the RAGE-deficient macrophages having higher levels of the cholesterol transporters, the cholesterol efflux to ApoA1 and HDL were significantly higher in the diabetic macrophages devoid of RAGE. So we wanted to understand what was the mechanism, because for the most part, the chief mechanism that regulates these transporters is through LXRs. And we cloned ourselves, actually, the promoters for ABCG1 and A1, and basically found that if we stimulated these cells bearing these reporter constructs with a RAGE ligand, this is one of the classical AGEs, carboxymethylysine, we were able to decrease the promoter activity and block that with a RAGE blocker. And then we looked down at the entire promoter, and we were very excited to find that the elements that appeared to be RAGE ligand RAGE dependent were not LXR dependent. And what it turned out when we did multiple mutations in various experiments to rescue, what we found was that the elements that were responsible for this effect were actually PPAR gamma dependent in the ABCG1 promoter, and that we could rescue this. So we don't quite yet know the full scale of the importance of this RAGE ligand RAGE dependence on affecting levels of PPAR gamma. But what we did with Dr. Fisher at NYU was laser capture microdissection of atherosclerotic mice. Here we actually, more lately, we've been using the LDL receptor knockout animal, and we generated double knockout animals with this as well. This is global deletion of RAGE. AGER is the gene name. And basically, when we laser capture microdissected the CD68 positive macrophages, what we found is that there were higher levels of the two transporters and also significantly higher levels of the PPAR gamma. And this, of course, may have a lot of implication, not only in this context with respect to cholesterol efflux and regulation of the cholesterol transporters, but also perhaps in macrophage inflammation signatures and as well in metabolism. And we do think that the future for our work now is really to try to understand how this RAGE pathway may actually function as a bit of a metabolic switch in these macrophages, and that this may direct their activity. And this became very interesting and important because many years ago, when I was still at Columbia actually, Tony Ferrante had been publishing his beautiful work about macrophages in obesity. And of course, it was very tempting to think about how we were discovering roles for the RAGE pathway in isolated macrophages and in atherosclerosis, to ask the question as to whether or not the function of RAGE in these macrophages might have to do also with the pathogenesis of obesity and not merely the complications when type 2 diabetes ensues. And so a very beautiful paper was published in 2014 in ATVB. And these researchers looked at human subjects, and they looked at the adipose tissue of lean and obese humans. And what they discovered was that there was a higher expression of RAGE in the obese adipose tissue, and that the ligands also were significantly higher. This is that same age ligand that I told you about before. And what this group discovered was that the RAGE was expressed in the macrophages that infiltrate the adipose tissue, in the vascular cells, of course, and obviously in the adipocytes as well. And so this work then was right smack in the middle of the work that we were doing in the mouse models. And getting together with Tony's group, we started to ask the question of whether or not the RAGE pathway might play roles in the development of obesity in mice fed a 60% high-fat diet. And so the first thing that we discovered, very similar to what the Gaines group discovered, was that high-fat feeding alone results in generation of the RAGE ligands. So we were very careful to look at the metabolic organs even before the animals became diabetic. So when the animals are on the high-fat diet for just a few weeks, they definitely don't have hyperglycemia and they don't have insulin or glucose intolerance. What they did have, though, in their metabolic organs was significant increases in at least two of the ligands. This is the AGEs and this is HMGB1, that inflammatory molecule that I told you about. Others have actually published that a number of the S100s are also increased in obese adipose tissue, and that's in the human as well as in the mouse model. So very importantly, we weren't looking at a complication of hyperglycemia, but perhaps we were looking at an opportunity to study the role of the RAGE ligands in the development of the obese metabolic dysfunction. So what was the hypothesis? So based on really that burgeoning literature at the time that we started this work, the hypothesis was that the homozygous RAGE null mice and their littermate controls would be fed the high-fat diet, and on eating this high-fat diet, they would become equally obese to the wild-type animal, but that they would be more insulin sensitive because of course, as you know, this very important relationship between the macrophages and insulin sensitivity had been being uncovered at the time. So very excitingly, that's not what happened. What happened was that the homozygous RAGE null mouse did not become obese on the high-fat feeding. So this, of course, led to many different sorts of experiments. Do they taste? Do they eat this food? And it was very, very clear when the animals finally went to an NIDDK-funded metabolic core led by Jason Kim that yes, they ate equal amounts of this food, but they did not develop the obesity. And so this, of course, was a big game changer, because obviously the relatively focused experiments in macrophages, metabolism, and inflammation was definitely not the explanation. So we also looked to be perfectly sure that all of the factors were aligned, and we looked at the insulin tolerance in these mice. Here we did insulin tolerance tests in our own lab, and we were able to show, looking at the area under the curve, that the rage-deficient mice, again, they were not obese, but in parallel, they did not have the insulin resistance that was observed in the wild type. And then Jason took the mice. And so he was able to get them up there, and these were all litter mates. And what he discovered, so essentially what they're able to do, of course, is many different things. And he looked at the VO2 consumption and VCO2 production. Mice, of course, are nocturnal. And what we discovered through this is that while eating the same amounts of this high-fat diet, these rage-deficient mice were able to increase their energy expenditure. And the same was true on the VCO2 production. This was the baseline, and also at 3 and 6 weeks of the high-fat feeding. So we also looked at the inflammatory phenotype, because we needed to be sure whether or not there was anything inconsistent with what we were seeing. And it was well described by Tony and others, of course, that in high-fat feeding, there's higher content in the fat of macrophages. And when you break them down to FB versus FBC macrophages, FBC also expressing the CD11C, which was believed to be more inflammatory, quote unquote, we found that yes, indeed, we saw the same thing with the high-fat feeding, but that these factors were all significantly lower in the high-fat diet-fed rage null mice. And so polarization is a very dicey sort of term. It's probably not accurate, but at least it's a start to be able to look at some of the known molecules that foster insulin resistance and others that are known to be protective against insulin resistance. And so we looked at the adipose tissue macrophages from the various groups of mice. And the bottom line is that the M1, the more pro-inflammatory macrophages, did not seem to be significantly affected by the deletion of rage. But in looking at 5 of these prototypical M2 type macrophages, all of them had significantly higher expression in the rage-deficient macrophages from the mice fed the high-fat diet. We actually don't believe that this has anything to do with the mechanism. So very preliminary studies in Affymetrix arrays, we have to repeat now with the RNA-seq, but in terms of what the Affymetrix array showed, it was astonishing. There wasn't a single inflammatory pathway. It was all metabolism. And so the bottom line is that the real question is, how does this rage pathway affecting these metabolic changes in these macrophages? And that's work that's still ongoing with labeling and all sorts of metabolomic work right now. But the bottom line is that it just proves that this concept really of this polarization may really not be the underlying linchpin mechanism in terms of how we get there. So the homozygous rage null mouse, it's very important to state that this is a very normal mouse. It develops normally. It has normal reproductive capacity. It has no obvious phenotype. It actually lives a bit longer than its counterparts. But in general, there's no obvious abnormalities. It still was very important, though, to use a distinct method to try to test this role in high-fat feeding in case we were missing some fundamental role of rage, for example, in adipocyte development, hypertrophy. And so what we did is we used the soluble form of the receptor, which is a ligand decoy. And for many years, while we were still working on getting that rage null mouse up and running, we used the soluble rage to give us a go, no-go on whether or not this pathway had anything at all to do with diabetes. And so in perfectly normal C57 black 6 mice, we began feeding them with the high-fat diet, and basically we allowed them to start to become obese here. And by the 3rd week, you do start to see differences in the body mass, and then we started either the soluble rage or the vehicle. And essentially what we found was that the rate in the increase in the body mass was lower, that the S-rage treated animals did not develop the same degree of the obesity as the vehicle treated animals, and they did have the same amount of food consumption, so they were eating the diet, and in fact they were more glucose and insulin sensitive compared with the controls. So this was an important experiment just in terms of being sure that there wasn't a developmental issue. So the really exciting part now that we're really happy to be pursuing is what is the mechanism of this? How is this RAGE pathway sort of suppressing energy expenditure on the high-fat feeding? So we started looking for clues, and here we looked at the visceral adipose tissue, and it turned out in the visceral adipose tissue, in the soluble RAGE-treated high-fat fed mice, that there was significantly more UCP1 expression compared with the vehicle-treated animals. We saw the same thing actually in the knockout as well on the high-fat feeding. And of course, UCP1 is a very important molecule in obesity and adaptive thermogenesis. And in cultured cells, what we discovered was that the RAGE ligands were able to downregulate UCP1, this is at the message level, and that this could be rescued with antibodies to the RAGE. And in very recent experiments, as I told you, that RAGE flox mouse is now finally back up and running and is breeding with the adiponectin Cree. We did a transplantation experiment where we put RAGE null brown adipose tissue into wild-type animals and then fed them the high-fat diet. So basically, this is using either RAGE null or wild-type brown adipose tissue transplants. And so far, the preliminary data appear that the animals that are receiving the RAGE null brown adipose tissue have a lower body mass compared to the wild-type. So of course, adipose tissue is really a conglomeration of many different cells that are in that fat. And the real question is what will happen with this flox mouse, and this work is ongoing. Evan Rosen actually gave his UCP1 Cree to the Jackson labs, and we've also been able to take a look at that, because the chief question here is whether or not this is primarily a Browning-Beijing mechanism, or whether or not it's a fundamental mechanism in the brown adipose tissue. We do know that when we put our homozygous regional mouse in the cold at 4 degrees, even up to 10 days, it very readily holds on to its body temperature compared to its littermate controls. So there's a fundamental mechanism by which this RAGE pathway is regulating this, and this probably is the bottom line reason why the animals are not becoming obese on the high-fat diet. And of course, those tissue-targeted animals hold the answer, to those important questions. And so Evelyn Litwinoff, she's a graduate student in the lab, has also taken on the project to determine and understand if in fact there could be a crosstalk. There's a very beautiful literature about how macrophages can crosstalk with the adipocytes, and it will be very exciting and important not to look at any of these things in a cell autonomous based mechanism. So Evelyn has been working in co-cultured systems using supernatants and cells that can directly adhere and touch, and basically we're really trying to be sure we cover all of the bases between cell autonomous and cell-to-cell interaction mechanisms to try to understand this. So for quite a number of years, I mentioned that we were very importantly focused on how the RAGE initiated signal transduction. We knew for many years that if we simply deleted the cytoplasmic domain but left a construct that could still bind the ligands, that what we had was basically a dominant negative effect when the ligands were introduced in either the cellular system or also in vitro experiments that we had generated, in vivo experiments with transgenic animals. But this is really did not really give us insight into how the cytoplasmic domain was able to engage signal transduction and activate these various pathways. And so in order to discover this, about 10 years ago, we performed a yeast-2 hybrid assay where we used the cytoplasmic domain of the RAGE essentially really as the bait. And then we fished into a lung library because the lung was where we discovered the RAGE from in the first place. And what came out were multiple clones of this molecule that turned out, based on the searching, to be DIAPH1. And so we were very intrigued, what is this DIAPH1? And it turned out that this DIAPH1 was a member of the Forman family. And so the Forman family molecules, they're very large molecules. They have many different functions that, at least initially when we looked at this, we were quite surprised how this could have anything to do with the RAGE pathway. But then, of course, once you start reading, thinking, and continue reading, you realize this may very well be an important mechanism. So it's extremely important to state that when these things come out of the yeast to hybrid, the very first to the 10th important thing to do is validate. And we used co-immunoprecipitation experiments with my colleague, Dr. Degatti at Columbia. We performed confocal microscopy. More recently, my colleague Alex Schekman has used FRET. And there's no question that this RAGE tail, the cytoplasmic domain, can interact with this Forman molecule, DIAPH1. So at least in terms of the physical interaction, those different techniques were able to show that this appeared to be a bona fide interaction. And what the Formans do is that they have two major roles, I would say. The first is that they appear to be important in actin cytoskeleton rearrangement, and of course this is important in cellular migration, which was a very well-known by many laboratories characteristic of the RAGE signaling mechanism. And also these formins are impacted by and they're effectors of Rho-GTPase signaling. So this is Rho-A, CDC42, RAC1. These are the cascades that can bring various signals into downstream signaling, and also, of course, activation of transcription factors that can then obviously do the important things of changing transcription and the cellular profile. And so based on these experiments, one of the very first things we did then was we asked, well, they do physically interact, and then we asked the important question of whether or not if diaphanous 1 is reduced in its expression in the cells, what will happen to the ability of the RAGE ligands to signal through the RAGE pathway. And so here, at that time, we used siRNAs, and we looked at the ability of the RAGE ligand to activate RAC1 and CDC42. And very importantly, when we knocked down the DIAPH1, we showed that the expression of RAGE was unchanged. So for some reason that we wouldn't have expected, all we really saw was that we were able to knock down DIAPH1, but we had no effect on RAGE. But what we did do is we blocked the ability of the ligands to signal as compared to the control siRNA experiments. So the next questions then were, well, what were some of the RAGE systems in which we knew that cellular migration and activation of Rho-GTPases was important in vivo? And we turned to a model system that we had studied actually quite a number of years back in the context of the RAGE in arterial injury. And here the arterial injury is induced by endothelial denudation in the femoral artery. We published a number of years back that homozygous RAGE null mice were protected from the aberrant neointimal expansion. And here, Art Alberts at the Van Andel Institute very generously shared his DIAPH1 knockout mice with us, and we tested this concept in this model. And basically, the first thing we always look for is, well, what happens to the expression of these various effectors once you institute the injury? And very similar to what happened in the context of RAGE, we found that after the arterial injury, the expression of the DIAPH1 was significantly higher, particularly in the smooth muscle cells. And importantly then, the real test of the hypothesis was if this increase in expression was related mechanistically to the neointimal expansion in these vessels, what happens to the DIAPH1 knockout mice? And so here we use the DIAPH1 knockout and its littermate control, and very similar to what we observed with the RAGE knockout mouse, we were able to show that the intima media ratio was significantly lower in the knockout. And so this slide really is a recapitulation of many experiments that we did in vitro in the smooth muscle cells because of that staining pattern that I showed you. And here, for the very first time, we were able to show that in the presence of the RAGE ligand, here we used S100B, and these were primary murine aortic smooth muscle cells, that the S100B could not signal all the way through to cell migration unless DIAPH1 was present. And we traced the signaling cascade to important roles for NOX1, generation of ROS, and serine 9 phosphorylation of GSK3 beta, which was critical for the formation of the lamellipodia, which is an important component of CDC42 and RAC biology, and that this stimulated then the cell migration. So these experiments then showed that this interaction could be the case. Now, what about in other systems? We have some preliminary data in human obese or mental adipose tissue that there's a significantly higher expression of DIAPH1 in the obese tissue versus the lean. In the mouse model, the same thing is true, very interestingly, especially in the adipocyte fraction from there. And also we know, oh, here it is, this is in the fat. And also we have started some experiments where we look at the homozygous DIAPH1 knockout mouse, and we're able to see already in just preliminary data that the high-fat fed DIAPH1 knockout appears to be at least partially protected from the high-fat diet-induced obesity. So those floxed mice also didn't have a good outcome in Sandy, and they're back up and running right now. So we're very interested to look at the adipocyte phenotype. In terms of diabetic complications, my colleague Vivette Degati at Columbia has really worked with us for many years to this moment, examining the expression of RAGE and now DIAPH. And Vivette actually was the first one to show that RAGE was highly expressed in the human diabetic kidney in the podocytes. And I remember we sent her over the DIAPH antibody. She called me very excited that in the diabetic people, the diabetic kidney of people, what she discovered was that DIAPH1 was also significantly higher expressed in diabetes versus the non-diabetic state, and she thought that the pattern was very similar to what happens in the case of the RAGE. So suggesting that this podocyte angle may actually be very important for RAGE DIAPH1 signaling. Some very preliminary unpublished studies, we know that we looked at the DIAPH1 knockout with type 1 diabetes, and preliminary data, Vivette has done all of the quantification, is that there appears to be less mesangial sclerosis in these animals. And she also sees that the podocyte effacement, which is very common in the wild type animals, this is after about 6 months of diabetes in the B6 background, that the podocyte effacement was significantly spared in her estimation in the knockout. She also found that the thickening of the GBM, the glomerular basement membrane, was less increased in the knockout compared with the wild type. So we have a lot of work to do now to try to understand. This is really a great opportunity to understand exactly how RAGE DIAPH1 may be involved in the pathogenesis of diabetic nephropathy. And using these various floxed animals, this really is a good start to being able to do that. So the last thing really that has been very important to us is that, you know, for these many years, it's definitely true that there is no RAGE inhibitor, you know, out there yet. And it's been a very challenging time. And I remember that in my years at Columbia, this was not really something that academic labs, you know, could try to do screening and all this sort of thing. Basically, they licensed it, and then essentially the truth is whatever would happen would happen. But the bottom line is that that's just not good enough. So right after the JBC paper about showing that the RAGE tail could bind to DIAPH1, I got a phone call from Alexander Schekman at the University of Albany, which was really just a big shift for us, and it was the beginning of a wonderful collaboration. And so Alex said, you know, I've been interested in the RAGE, and you must have seen some of my papers. Alex is an NMR spectroscopist, and he was very fascinated by the extracellular domain. And I remember he said to me, he said, you know, but that extracellular domain is so complicated. I told you about the various regions where these ligands combine. And he said, let's work on the tail together. And so wonderfully, what Alex discovered, he said it's much easier to work with. You know, this is only 43 amino acids in the human as opposed to 332 in the extracellular domain. And lo and behold, Alex was able to solve the structure of it. And Alex proved that these 2 amino acids in the RAGE tail, R5Q6, he showed through many different ways in his NMR spectroscopy and biochemistry techniques that these amino acids were absolutely essential for the RAGE to bind to the DIAPH1. And then in our lab, we took those same constructs where we mutated R5Q6 to alanines, and what we were able to show is that in the presence of the double mutant, just like Alex showed that the mutants no longer bound the DIAPH1, what we were able to show was finally a much better experiment than just deleting the cytoplasmic tail. Because in the presence of those mutations, everything else was present and equal, except R5Q6 were mutated to alanines. And what we were able to show, this was in smooth muscle cells, was that the RAGE ligands, here we used S100B, were not able to signal through phosphorylation of AKT. We also looked at functional experiments in the smooth muscle cell, and the mutant was able to block the ability of the ligands to stimulate proliferation and migration of the smooth muscle cells. But really importantly, what we did not find was that the mutant had any effect on non-RAGE ligand activation. So for example, we used PDGF, which is a very potent stimulus of proliferation, migration, and the mutant had no effect on this. Now, of course, this doesn't mean that this is specific. We still have a lot of work to do to sort it out, but it was encouraging that this may be a very specific component of this biology. So together with Alex, what we did is we developed a high throughput binding assay in our laboratories. And essentially what we do is we use an antibody to immunoprecipitate DIAPH1. And then we purchased a library. Actually, we bought this library. 3 investigators from Columbia split this library because it's really very ridiculously expensive, to be honest, but what can you do, you know? It was important, so 3 of us went in and bought this library. And so each of us had a daughter, and the bottom line is it had 59,000 compounds in it. And in the 59,000 compounds, Alex and I worked out a way to be able to just go back and forth on trying to understand whether or not they bound to the RAGE tail. And he had to use his biophysics. We did all of our cellular-based studies. And the bottom line is we went from 59,000 compounds, and we got it down to 14. And so from those 14 then, these now are the basis of putative platforms for developing potential therapeutics. So in the laboratory, we tested them in all different types of cells. And the bottom line is that the final experiment was an experiment that recapitulated the exact same experiment that I did maybe 20 years ago with the soluble receptor. And what we do in that experiment is we inject the RAGE ligands into the mice, and then we treated them with the compounds. Basically, the compounds fall into probably multiple different series. They're not 14 individual molecules. Many of them are related. And we found that when we pre-treated the animals with these 2 different classes of the molecules, these were from the original library, that we were able to significantly block the pro-inflammatory effects of the RAGE ligands looking in the liver and in the kidney tissue. So of course we know that we have a long way to go, but we've been very fortunate. NYU has a wonderful mechanism for helping the investigators to do translational approaches in their work. And they brought in a medicinal chemist, Dr. Bob DeVita, and Bob actually has really been a critical person in understanding these molecules. Together, myself, Bob, and Alex Schekman, again from Albany, are doing what Bob calls structure-activity relationship refinement, meaning making them better and better and better. And it's just a constant interplay and interaction between what parts of these new molecules work, what doesn't work, and hopefully they're platforms, you know, for therapeutic development to try to get this, you know, to the next step. So in summary then, I think I've told you what some of the updates are of where we're at. And we're pretty, you know, excited about now some new possibilities for finally understanding the natural function of the receptor, perhaps as an energy hoarder and retention and energy, and obviously very exciting pathways in terms of macrophages and their roles in lipid metabolism and cellular metabolism. And then, of course, it's really important to continue to understand how the molecule signals, what are the implications of this. And so these small molecules that we're developing, obviously, are really very wonderful probes now to be able to try to understand the signaling aspects, you know, much better. So in closing, I would like to acknowledge all of the present and past members of the laboratory. And I told you about Alex Schekman, Art Alberts, who shared those wonderful mice with us, the DIAPH knockout, Tony for all the work continuing to this day in obesity, Richard Friedman, who did the bioinformatics and was the first one to discover that the RAGE deficiency appeared to upregulate those cholesterol transporters. He found that, very, very readily in the work that he had done. And also, of course, I mentioned Dr. DeVita, the medicinal chemist with whom we are working to try to optimize the small molecule program. And lastly, we're very grateful to the funders for, having continued faith and let us keep going. So thank you very much for your attention. It was great to be back. Thank you.
We have time for some questions.
Hey, Marie, maybe I misunderstood this, but when you fed the mice a high-fat diet, and then you didn't have the weight gain and you had increased energy expenditure, wouldn't you expect the adiponectins to increase, not decrease, or is there some kind of interplay with the adiponectins receptors?
Adiponectin was higher in the RAGE knockout. Yes.
It was, okay.
Yes.
Yeah, I thought it decreased.
Yes, we looked at that, yeah, absolutely, and it was significantly higher. That's actually, I didn't highlight that, but that's also one of the reasons why we're interested in that crosstalk, to try to understand, who's talking to whom, if they are, the macrophages and the adipose cells. Absolutely, yes.
As you know, with normal aging, there's a decreased sensitivity to insulin.
Yeah.
I was wondering what you found with normal aging and RAGE changes.
Yeah, so what we had done actually is we took the mice to about 24 months of age. And the honest truth is that in the C57 Black 6 background, we were not able to see big differences in insulin tolerance. So it's possible that we would have to have let them get older. We did try to look at that, but even at that age, we were not convinced that their ITTs were different. Now it's possible, I suppose, to do the hyperinsulinemic euglycemic clamp on them, but that is quite frankly something that we did not do.
I'm wondering whether when you looked at compounds that could decrease the RAGE inflammation, whether you looked at some cheap generic drugs such as aspirin, which has an effect on inflammation, such as statins, which have inflammatory as well as lipid antagonism.
In the screening, yeah, actually that's the first thing you're always advised to do exactly that, to buy these little panels. And in the screening, those molecules had no effect, at least on the physical interaction of the RAGE with the DIAPH. It is known, though, that from work that others have done, that humans treated with statins and also I think with the TZDs, that they show in some of their accessible PBMCs, expression of the receptor actually is decreased in the diabetic or the atherosclerotic state. So that is, others have shown that that's one of the benefits potentially of statin and TZD treatment in the human, but it doesn't affect the binding specifically.
Fantastic story. I was wondering, I may have missed it, but is there actually expansion of the brown fat in the mice?
That's a very good question. So the story is that if you weigh the fat in the knockout, it doesn't appear, this is the brown fat, it doesn't appear to have a higher mass. We have a few mice where my colleague Yushin Ding, who has done PET imaging with FDG, and she's showing that in the RAGE knockout, even though the mass is the same, that the FDG uptake is significantly greater in these mice on a high-fat diet. So what you have hit on is exactly the real problem that we face now. We have to now look, so that's the brown adipose tissue. The next step is the subcutaneous adipose tissue, which is really where the brown and beiging occur, and then also on the third depot, where it's less, is in the visceral adipose tissue. Believe it or not, what we're seeing is that those sites vis-a-vis the RAGE DIAPH1 are not equally affected by the cold room and by the high-fat feeding. They appear to be behaving a little differently. So we have to dissect these out individually. At least in the brown, there is no direct expansion. It appears to be activity.
So do you see greater decoupling in the mitochondria?
Yes. Yes, in the seahorse, absolutely. Totally unexpected, Because now with the seahorse, you can take these cells directly from the animals, and plate them, and it's absolutely decoupled. Yes. The question is, why? And this is what we're really working hard on trying to understand. It's not directly at transcriptional regulation of UCP1. We just finished experiments where we took the promoter apart, and there's absolutely no effect. It's upstream, and it's just a little elusive right now what the mechanism is, but it's upstream of a direct transcriptional regulator.
Great. Let's thank our speaker for.
Thank you.